when you're selecting an antibody, you often want to find the application that it was used in that particular application. Of course, it's more powerful if you see data. So at R&D Systems, and bio-techne, when we make antibodies, of course, we have to screen. We literally screen them against all of these techniques. And of course, to be able to release them commercially, they have to perform. There has to be a high signal, and it has to be low noise. It has to be reproducible. And those that don't pass muster, they don't get released. And what works for one application will not always work for another, obviously. So a couple things to think about, and I just want to take a few seconds on this. Of course, the first credible place that people look, of course, are publications. This is the obvious thing, and it's a real credibility statement when your colleague uses an antibody. But the second thing that I wanted to mention, which is, and we mentioned the word trust very early on in this talk, and you really should evaluate a vendor based on what they do to make that antibody. I'll give you one example. When making a polyclonal antibody, are they using a peptide or are they using a full-length, properly folded protein? Which is what we do because we make proteins. So that is something, because you want the most natural epitope state for that antibody to recognize, and not using peptides. Things like peptide blocking to assess specificity. And then there's one issue that has come up, I think, recently in the scientific community, and I think there's been some articles about this, and this is the whole idea of reproducibility and consistency. Because my gosh, if you run a blot on day one with an antibody and you can't reproduce that the second day, and a lot of papers, there have been some papers that have come out that have been questioned based on their reproducibility. You've got to be able to show lot-to-lot reproducibility with an antibody. It's really, really key. And this is internal data that this is actual data from our labs at R&D Systems bio-techne that shows that from lot to lot we get the performance exactly right every time, perhaps plus or minus 10% CV. And this is a really important thing because you want to be able to trust that antibody in the long run, that it's going to perform lot after lot and you don't get any kind of drifting. Now, I want to show you some more powerful data. We talked very early on about lysates and sample prep. And this is a very underlooked area. And I got a lot of this data, by the way. I'd like to acknowledge Jeff Cooper, who is from our labs at bio-techne, R&D Systems, who's in, I don't know, Jeff and I just met each other recently through emails, and he basically works in our antibody development group. And I thought some of this data was quite powerful because what it basically says is it's not always the antibody. We're always quick to judge the antibody. But again, garbage in, garbage out. So this is a very simple experiment that shows that, so what we did here is, and what we do is we screen, before we even screen antibody clones, we have to screen lysates because we need a optimal top performer lysate to then be used to then screen, say, our antibody clones. And so these are exactly the same treatment. These Dowdy cells are treated with interferon alpha exactly the same way. There were 2 different technicians involved, but the passages, they were collected at different passages, passage 42, 31, and passage 5. And what this shows is, this is a phosphostat-3 antibody, that you're getting different pre- and post-induction signal. And so, the question is, well, why? Well, there's multiple variables, and we started to talk about this. And I guess my overall message is you want to be consistent. You want to have a good positive control. You want to be consistent in your pipetting. You want to make sure your phosphatase inhibitors and your protease inhibitors are fresh and you're using the right ones. You want to minimize your freeze thaws. You want to, make sure your cell density is not too high and you're collecting the cells at the right passage number, and your reagents are fresh. And even, if you think about it, the protein quantitation assay you're using. So, you could be using a BCA, a Lowry, a Kamasi Blue, or, nanodrop, UV-based, and they all work based off amino acid content. You want to make sure you're consistent there because you can get different, total protein. And the point, the message of the slide is that don't always blame the antibody. Pay attention to your lysates. Another example, same exact conditions across. This is a phospho-FAC antibody, and showing exactly the same treatments, exactly the same cells, and we're getting the pre-induction is all very clean, but the post-induction gives you varying signals. And the question is why? Well, there's variations here potentially, and this was actually used to screen, so they're doing the pre-screen in the lysates to then be used to screen clones. if you were to use lysate from lane 1, you might underestimate the power and the reactivity of that clone versus lysate 2, which shows a much stronger signal, and you might miss some clones that could have been very, very well, that could have been very, had great affinity and great avidity, versus using lysate 1. So you want to make sure your lysates are prepped properly and you pay attention to all the small details here. Another example, quickly, this is HeLa cells, PMA treated. Again, same conditions, just more data to reemphasize the fact that, identical treatments do not always give identical results. Pay attention to your lysates.
To view the full video of this and all of our other webinars for bioscientists at the bench, please visit bitesizebio.com/webinars.